All right, let's talk about something that I know can feel, well, a little intimidating. The IB Biology Internal Assessment. It often seems like this giant mountain you have to climb. But I promise you, by the end of our chat today, you're going to have a complete blueprint. We're going to break it all down, step by step, so you can turn this thing into a really cool, totally manageable scientific adventure. Ready? Let's get into it.
First things first, why should you really care about this? Well, look at that number. The IA isn't just some small assignment. It is a massive 20% of your final IB biology grade. That is a huge chunk. A great IA can seriously lift your overall score, so yeah, it's absolutely worth putting in the effort.
So what is this thing, really? Think of it this way. The IA is your shot to be a real scientist for a little while. It's your original investigation. You get to call the shots. You come up with the idea, you design the experiment, you gather the data, and you figure out what it all means. This isn't about memorizing flashcards. It's about putting your critical thinking and problem-solving skills to the test.
Okay, so keeping that big 20% in mind, how in the world do you even start a task this big? Don't worry, we're going to break it down into a super clear, logical process, and we're starting with what is, without a doubt, the most important part of the entire project.
You know, every single great scientific study and every high-scoring IA is built on an incredibly solid foundation. And for you, that foundation comes down to two things: a great research question and a solid hypothesis. If you get these two things right, I promise, everything that comes after gets so much easier.
So it's basically a two-step process to build this foundation. First, you've got to craft that perfect, focused research question. Then, you use that question as a launchpad to develop a really clear, testable hypothesis. Let's dig into what makes a research question really good.
There are kind of 4 golden rules here. Your question has to be focused and measurable. So not how do plants work, but something way more specific. It has to be something you can actually test in your school's lab. And this is a big one. It should be genuinely interesting to you. You're going to be spending a lot of time on this topic. And finally, the best questions usually start with what or how.
Here's a perfect example. What is the effect of water's pH level on plant growth? See how that works? It totally hits all the marks. It's specific. We're talking pH and growth. It's measurable. You can easily measure pH and track how tall a plant gets. And it starts with what? This is the kind of strong, focused question you should be aiming for.
Now, once you've got your question locked in, you make a prediction. That's your hypothesis. It needs to be a super precise statement that your experiment can either prove or disprove. So using our example, a solid hypothesis would be something like water with a more neutral pH will contribute more to a plant's height. It's simple, it's clear, and it's totally testable.
Okay, with a solid question and hypothesis, it's time to actually design the experiment. This is where you put on your serious scientist hat. It's not just about mixing some stuff together, it's about being incredibly careful in controlling everything so you can get data you can actually trust.
A flawless experiment design is all about defining your variables. You've got 3 types to figure out. First, your independent variable. That's the one thing you're changing on purpose. So in our example, it's the pH of the water. Then there's the dependent variable. That's what you're measuring to see the effect. In this case, the plant's height. And finally, you have your control variables. This is everything else you have to keep exactly the same, like the light, the temperature, the soil, all of it.
And speaking of controls, you absolutely 100% need a control group. This is your baseline. It's what you compare everything against. For our little pH experiment, this would be a plant that just gets regular, neutral pH water. Without that group, you have no idea if your changes actually caused anything to happen. It's non-negotiable.
All right, next up, the number 3. Why is this number so important? Well, to make sure your results aren't just some random fluke, you have to do your whole experiment at least 3 times. This is called replication, and it's fundamental to good science. It shows that your findings are consistent, and trust me, it's a huge deal for getting a good score on your IA.
So, you've run your experiment, you've done your repetitions, and now you have all this data, pages of numbers and notes. This next part, the analysis, is really the heart of your IA. This is where you take all that raw information and turn it into real, meaningful scientific evidence.
Now, you're probably going to have 2 kinds of data. Quantitative data is all about the numbers, anything you can measure, like height in centimeters or a heart rate in beats per minute. Qualitative data is more descriptive. It's your observations, like changes in leaf color or texture. Both are super valuable, so make sure you're recording everything.
But just having a list of data isn't enough.
you have to analyze it. This means doing some basic stats. You'll definitely want to calculate the average of your results, figure out the standard deviation to see how spread out your data is, and most importantly, look for the trends. What is the story that your numbers are trying to tell you?
And that brings us to the final piece of the puzzle, writing the actual report. This is your chance to communicate everything you've done. I like to think of it as telling the complete story of your scientific journey, from that very first question all the way to your final conclusion.
The good news is, the structure is pretty standard. You'll start with an introduction that lays out your research question and hypothesis. Then a method section explaining exactly what you did. The results section is where you show your data with tables and graphs. The discussion is where you interpret what it all means, and finally, you'll have your conclusion and bibliography.
Through it all, clarity is key. Here's the goal you should aim for with your methods section. It needs to be so clear and so detailed that another student could read it and repeat your experiment perfectly without ever talking to you. That right there is the gold standard in scientific writing.
So before we finish up, let's just zoom out for a second. Why are we going through all this trouble? Because this project is so much more than just a box you have to check to get a grade. It's really about building skills that you're going to use for years to come.
And the possibilities for what you can study are just endless. To get your brain going, think about some of these. How does music affect your heart rate? Do natural pesticides actually work? You could see how pH affects yeast, or compare different soaps, or even test how light color impacts photosynthesis. Just find something that makes you go, I wonder.
Seriously, the long-term value here is incredible. When you finish this IA, you're walking away with real problem-solving skills and university-level research abilities before you even get to college. If you're thinking about a career in science or medicine, this becomes a fantastic piece for your portfolio. But more than anything else, you are learning how to actually think like a scientist.
So, there you have it, a complete blueprint for conquering the IB Biology IA. We've walked through the whole process, from coming up with that initial question to writing the final report. The path is all laid out for you. So the only real question left is, what will your scientific adventure be?